Today we'll be starting with transposable genetic elements, and the books I have referred is Molecular Biology of the Cell by Lorish, Molecular Biology of the Gene by Watson, Snowstad and Simmons Genetics, Broca's Genetics, Russell I Genetics, Albert Cell, and Lewin Gene 11. Now, when we consider transposable genetic elements, what does transposable genetic elements mean? By definition, if we go, these transposable elements are mobile DNA sequences. That is, DNA sequences that can move within the chromosome, between the chromosome, and are found in genomes of almost all organisms. For example, in humans, at least 45% of human DNA seems to have transposable genetic elements. Now, as we understood that these are DNA sequences that can move. So in most cases, transposable elements are able to insert themselves at many different locations, rely on mechanisms that are different from homologous recombination. By inserting into different sequences or different areas of the chromosome, they may cause mutations either by inserting into another gene and disrupting it, or by promoting DNA rearrangements such as chromosome deletions, duplications, and inversions. So from the first slide and whatever I told now, we have just this basic idea that these are DNA sequences that can move. Now, coming to this lady, this is Barbara McClintock, who was awarded the Nobel Prize in 1983 for her lifelong work on maize. And she was very passionate about her work. Now, Barbara McClintock began her scientific career at the Cornell University, where she pioneered the study of cytogenetics in 1930s using maize as a model organism. Now, how do we relate transposable genetic elements to Barbara McClintock? It was her lifelong study with maize that finally gave the scientists insight into what are these mobile DNA elements. In 1931, she and another graduate student, Harriet Creton, first provided evidence that genes were physically positioned on chromosomes, and they described the crossing over phenomena and genetic recombination. As I said, she is famously known for her discovery of transposable elements through experimentation with maize. And when at the beginning, she had informed the scientific world that there are DNA sequences that can move within chromosomes, between chromosomes. That means DNA can move. That was basically the whole scientific world was taken aback, because the time we are talking about, 1930s onwards, so it was early 1940s, 1950s, you can understand the DNA was being discovered. DNA was known for its stability. Then how is it possible that a sequence of DNA can jump from one chromosome to another chromosome? So when she described her work at the initial instance, as I said, everybody took it critically. However, later on, as she explained, and with the discovery of the DNA structure, further proofs coming from a variety of scientists, conclusively it was proved that she had a big hand in establishing the fact that sequences of DNA can jump, a stretch of DNA can jump. And for that, she was awarded the Nobel Prize in 1984. So this is Barbara McClintock. And now we are moving on to transposable elements. When we classify transposable elements, they can be cut and paste transposition. I'll come to them one by one. In your syllabus, you have this cut and paste transposition, and we will be studying the IS elements. We will study one composite transposon. We will see the ACDS element. The ACDS element was the element on which Barbara McClintock worked. That was her work. We will also see, have a look on the P elements. The P elements are found on Drosophila. And we just have a basic structural idea of the replicative transposons. Now, automatically, you people might be having a question as why one are known as cut and paste, the other replicative. So I'll be showing it to you in the next slide by diagram. So the first category is cut and paste transposition. The second category is replicative transposition. So we will be studying TN3 elements also. And the third category is retro transpositions. That is, here stretches of DNA do not simply come out of the chromosome and move. Stretches of DNA, which are considered as retro transposons, move via RNA intermediate. How is that possible? We'll see it by the next slide. So we'll not concentrate on retro transposons much because they are not a big part of your syllabus, but for a glimpse, we'll understand what are retrotransposons also. And if we look into retroposons, retrotransposons are of two categories, retrovirus-like elements and retroposons. Of the retroposons, we will study these two elements, the lines and the signs, which are found within the human genome. So this is what we will be studying about the transposable genetic elements. So as I said, the cut and paste transposition, the replicative transposition, and the retrotransposons. So initially, before entering into the details of transposons, we must keep it in mind that there are three basic categories: cut and paste, replicative, and retrotransposons. Now coming to the transposition phenomenon. As I said, transposable elements is a piece of DNA that can move from one location to another location in the cell's genome. So if this is the DNA sequence that is present here, it can move out to another region within the same chromosome or into a region of a different chromosome. If you look into this picture, one thing that is notable is that when it is moving out, it is not keeping a copy of itself. In this case, what they have shown is that it is moving in the same chromosome. It is moving from a place into another position in the same chromosome. And if you look here, when it is moving out, it is not keeping a copy of its own chromosome or own self. So these are basically cut and paste transposons. Let's look into the next part. This is another transposon which is moving. And as you can see, so in this case, as you can see, this is the DNA sequence that is about to jump. But while jumping, you can assume that, just a second, it is keeping a copy of itself and then jumping into a new position. So this is replicative transposition. So when I discussed the three basic types of the two basic types of DNA transposon in the previous case, where the DNA jumps, I said that there is this cut and paste. So we understood that cut and paste is the movement of a transposon to another place, not keeping a copy of itself, while the replicative transposition is, it is keeping a copy of itself and it is then moving it into another position. And finally, we have the retrotransposition that we just heard in the previous slide. So in the retro elements, as you can see, this is the sequence of DNA that is supposed to jump, but they cannot jump. What they do instead, they make a RNA copy of themselves, these DNA sequences, which are retro elements. And this RNA copy, using the cellular reverse transcriptase, makes a DNA copy. And this DNA copy thus integrates into the genome. Now look at the output. So we have the original retro element in its position because it's there, it didn't come out. It just moved through RNA intermediate, and we are now having a fresh copy of this same retro element. So thus summarizing, we can say that the transposons or DNA sequences can jump within the chromosome, between chromosome, from plasmids to chromosome. Plasmids are circular DNA molecules found in certain bacteria. It can jump between plasmids. And again, another kind of definition I have ordered the group of this jumping elements into two basic groups: insertional sequences and transposons. What we saw here are basically transposons. Then what are IS elements? When we look at the structural construction of the DNA sequences that can jump, those DNA sequences which have a very simple system are known as insertional sequences. I'll show you by diagram. And those which have a larger size is important. Insertional sequences size are smaller. While in case of transposons, the DNA sequences that are known as transposons, or the stretch of DNA that is known as transposon, the size is comparatively much, much larger. So if we define it this way, the IS elements are relatively small, with a size ranging from 560 to 2500 base pairs. Well, transposons are a varied category. One transposon known as TN10, transposable element 10, it has a size of around 9,300 base pairs. So you can understand, insertional sequences are simple and smaller. Transposons are more large and complex. When we'll see the diagram, we'll have a more clearer picture of what is the difference between insertional sequence and a transposon. However, just remember, both are sequences of DNA that can jump. So we did that. Now, as I told you, simple transposition. Now, so it is it's cutting itself and inserting, cut and paste. It is inserting itself into another position. And look at the structure of a simple transposons. Simple transposons means those DNA sequences which are cutting themselves and inserting themselves into any other location within the same chromosome or into a different chromosome. Under simple transposition, we have these two basic categories of transposable genetic elements. One is the insertion sequence. As you can see, the structure I said, the complexity, it's not much complex, it's much simpler. And if you look into the composite transposon, you can see that the size is bigger and there are a number of elements that has been introduced into the composite transposition. For information and for studying this picture, I'm just writing here that there is this DR. DR means direct repeat. And another thing you can see is that IR. IR means inverted repeats. So direct repeats, how do we read it? If it's 5' to 3' direction, so a direct repeat is T, C, G, A, T. In the same strand at the 3' end also, we will have a T, C, G, A, T. So that is a direct repeat. Just a second. Okay, let this be read. So if there is a direct repeat here, that means if the sequence is read as TCGAT, then there is another direct repeat here. The sequence is read as TCGAT. When we are talking about inverted repeats mean if in the 5' direction, the direction is important. In the 5', 3' in one strand, if the sequence is, for example, this is a simplified example, ATG, then in the opposite strand, again in the 5', 3' direction, we will have ATTG. So this is the concept of direct repeat and indirect repeat. When we are thinking of direct repeat in the same strand, in the same orientation, the sequence is same. When we are thinking of inverted repeats, it is a two opposite strand and the sequence in the 5', 3' orientation is same. So these are the two terms that has been introduced in the picture. And let us start with this one. This is the insertional sequence. You can see that if this is the, just a second, let me remove all this. If you see, this is a simple DNA sequence that has the capacity to jump. Now, the sequence that has the capacity to jump should have a gene that will facilitate its jumping and recombination or insertion into a new place. So that gene that facilitates its jumping and helps in insertion and recombination into the new place is the transposase gene, which is a recombinase. So if the presence of the transposase gene and consequently the presence of the transposase product facilitates the jumping of these DNA sequences. In contrast, if you look into the simple structure of a composite transposon, you can see there is a direct repeat, there are inverted repeats, at the end again there is a direct repeat, there are inverted repeats, and apart from the transposase gene, it has another gene. It can have one or more genes. Here, for example, they have shown that there is another gene, which is the antibiotic resistance gene. So this is about simple transposition. As we move to the next, now we have replicative transposition. If you see the replicative transposition, as we discussed, it is keeping a copy of itself and moving into a new position. And look at how the replicative transposons look. If you compare it with a composite simple transposition, there is this addition of new gene, which was missing in the above two, that is the resolvase gene. So in case of replicative transposons, it needs to keep a copy of itself and then move to another part. So there is this co-integrate form and there's this specific structure forms that results in this phenomena. So I'll be showing that structure to you while through Watson's molecular biology of the gene animation. So I'm not getting into details of how that happens here. I just wanted to show you in comparison that in case of replicative transposons, we have this additional enzyme which is needed for these molecules, these stretches of DNA to keep a copy of itself and move into a new position. Next, if we move, and the third category which I told you was a retrotransposition. Now, if you look, so retro elements, as you can see, they have an RNA intermediate, and the RNA then makes a DNA copy by the enzyme reverse transcriptase, and then the DNA is integrated into the new position. So if we look into these elements, you can see that apart from having the reverse transcriptase gene, because this is essential, as you can see, the DNA itself is not coming out. So the need of transposase gene does not arise, but the DNA should have a reverse transcriptase gene so that the RNA can make a DNA copy of itself. So it is having the reverse transcriptase gene and the integrase gene, which helps in integrating the complementary DNA. Now, there are two categories of retrotransposons. One was the viral-like retro elements. As you can see, there is this direct repeat. There is this another element known as long terminal repeats, which are found in viruses. And then again, there is this direct repeat. In humans also, we have non-viral like retro elements. That means these DNA sequences present in the humans move through RNA intermediates. In case of humans, it is much simpler. They have a direct repeat. There is this reverse transcriptase gene, and again, there is another direct repeat. So this is a comparative structural analysis of the variety of DNA transposons and the retrotransposons that are observed in nature. So I have brought the three slides together. So you can understand one thing: for retrotransposition, the transposase gene is not required. But when it is a DNA transposition, DNA is moving, the transposase gene is required. If the DNA is moving in a replicative fashion, then an additional gene, resolvase, is required. So that was a comparative analysis of all the three categories of transposable elements that are observed. Now let me come back again to the concept of direct repeats and indirect repeats. Here, as you can see in this picture, they have shown, sorry. In this picture, as you can see, they have shown a transposable element. See the strand, note the strand. One strand is moving in 5' to 3' direction. The other strand is in the 5' to 3' direction. Now, look at this repeat sequence. It reads TGCAA. And you can see on the 3' end also, it reads TGCAA. So the direct repeats, when we read the same strand in the 5', 3' direction, on one end it is a TGCAA, on the other end also is TGCAA. Now coming to the other thing that I just discussed while explaining the other slide was the inverted repeat. When we read the inverted repeat, we always read it in the 5', 3' direction for one strand and the 5', 3' direction for the other strand. So if you read the sequence here, it is ATCGCA, 5' ATG, sorry, 5' ATCGCA 3'. Again, let's read it here from this end. It reads 5' ATCGCA 3'. So all DNA sequences that are capable of jumping are flanked by inverted terminal repeats or direct terminal repeats. For ease of diagram, the inverted terminal repeats are represented in this manner in books, and the direct terminal repeats are represented in this manner in your textbooks. So with this basic concept that what is a direct repeat, what is an inverted repeat, how a direct repeat is written in textbooks, shown in textbooks as I've showed you here, and inverted repeat shown in textbooks, to keep in mind, obviously at the back of the mind, you have to keep that all DNA sequences cannot jump. Only some DNA sequences can jump. Those DNA sequences that can jump has some properties. So one of the property of these DNA sequences that are able to jump is that on the both sides, at the both ends, they are flanked by inverted terminal repeats and direct terminal repeats. That is basically a criteria of transposon, usual. That is the usual criteria of a transposon. Now, question is again that the DNA can jump. And we have understood by now that there is one mechanism of jumping. You cut yourself and you paste yourself in another position, or there is a mechanism of jumping where you keep a copy of yourself and paste yourself into another position. So this is a very simplified diagram. I'll be showing you this mechanism of cut and paste mechanism of transposition by animation also, but this is a diagrammatic representation of how this happens. Now, the site where the transposon wants to get itself integrated, remember, the DNA is coming out and it will now insert itself into another place. So let us assume that DNA to be the target DNA. In the target DNA, staggered cuts are made. Staggered cuts mean, if you look into this arrow, one strand is cut here and the other strand is cut of around 2 to 9 base pair gap. So the two strands, as you can see, one is cut here and the other strand is nicked here. So if you, and then all the hydrogen bonds between these two, the bases are also nicked off. If something like this happens, automatically the two DNA will open up. So as you can see, as a result of this, the two DNA molecules have opened up. The double helix has opened up. And our transposable elements now insert itself into this position. Once this position, once the transposable element has inserted itself into this position, what happens? We are having a single-stranded DNA here and a single-stranded DNA here. Taking this to SS DNA as the template, the 3' end of this 2 DNA are then filled in. So you can see, as a result of which we are getting the 2 direct repeats flanking the transposon. So this is how, it's in a very simplified diagrammatic manner, I've shown you how cut and paste transposition occur. Obviously, how the original transposon cut itself out from the DNA has not been shown here, but how it gets inserted into a new place has been shown in this diagram. Now, when we are talking about, this is another picture showing you the 2 categories of transposon based on whether they are found in eukaryotic DNA. So if you look into the eukaryotic DNA, they are basically the DNA transposons. So there is a donor DNA, there is a target site cleaving, donor DNA inserted. If it is a retrotransposon, it's this is the donor DNA. The donor DNA is making an RNA. The donor DNA doesn't come out. And then a cDNA is made, target site recognized or target site identified, and this DNA is then inserted. So this is a very simplified mechanistic approach of how the donor, from the donor DNA, in case of DNA transposon, the DNA can come out itself and get itself inserted or integrated into the target site. And for retrotransposon, the donor DNA do not come out from the main original position. It makes an RNA copy from which a cDNA, the cDNA is integrated into the target site. So by now we have understood, we have got this basic concept of cut and paste transposition. We have understood replicative transposition, and we have understood retrotransposition. Now, when we again classify transposition from the aspect of whether they are viral-like or they are DNA transposition, they again are of 3. By examples I've shown you, they are again of 3 basic categories. When we are talking about DNA transposons, there are 2 examples given here, TN10, TN3. When we talk about viral-like transpositions, that is, they move by RNA intermediates, but they are viral-like. These includes the retroviruses. And there are some which are known as retrotransposons. And these elements are non-viral transposons. And in case of humans, we saw in one picture, we will be studying the example of lines and signs. Now coming back to from where we started. In one of the slides, I showed you that based on the complexity, how large the DNA sequence is and the size, structurally, there are 2 basic classes of transposons. So the first one is insertional sequence. These are the simplest of the transposable genetic elements. They have a central DNA piece which contains the transposase gene, which helps in moving of this DNA sequence from chromosome to chromosome or within the chromosome. And this central piece of DNA is flanked, as you can see, by 2 inverted repeats. So when this transposon will cut itself out and integrate into a target site, automatically this transposon will create a direct repeat on this side, as I just showed you in a very simplified manner. That is how direct repeats are generated when a transposon is integrated into a fresh site. So this is the insertion sequence, the simplest of the transposons. Now coming to the transposons. As I said, they are much larger in size, and you can see it can have a number of genes. For example, it obviously has the transposase gene. It can have the resolvase gene if it is a replicative transposon. It can have certain antibiotic genes. For example, this is the tetracycline resistance gene. Apart from that, this is something to be noted. See, the borders are flanked by IS element. So a IS element, one IS element is present here and the other IS element is present here. So if the central piece of DNA doesn't have the transposase gene, it doesn't matter because the IS elements, so there is this IS element present on the left side of the central piece of DNA, which is known as the IS10 left. And there's another insertional sequence present on the right side, which is known as IS10 is a name given to it. Insertional sequence 10 on the right. And any one of the IS element can provide the transposase, both of them have the transposase gene. So any one of them can provide the transposase enzyme for this whole transposon to jump out. So this is a comparative analysis to show you that IS elements are basically very, very smaller structures. They have only the transposase gene and are bordered by inverted repeats. But when we talk about transposons, they are large pieces of DNA. And they also have boundaries. In this particular picture that you are seeing, TN10, the boundaries are bordered by two insertional sequences, which any one of the IS can provide the transposase enzyme for this particular transposon to come out of the DNA and integrate itself into a new area, new region of the chromosome. We have understood the basic conceptual difference between insertional sequence and transposable transposons. Now the question arises that are all transposons bordered by IS elements? No. Some transposons have insertional sequence at their boundaries, but it is not necessary that all transposons will have insertional sequences on their boundaries. Based on the presence or the absence of insertional sequences at their boundaries, we now can differentiate transposons into two basic categories. One is the composite transposon and other is the non-composite transposon. Now look at the picture. The picture speaks of itself. You can see this is the central piece of DNA. This is the central piece of DNA. In this case, the DNA is bordered by two IS elements on the two sides. So this is a composite one. The central piece of DNA might not have the transposase gene, but any one of the IS element, each of the IS elements have the transposase gene, and they can produce a transposase enzyme, as a result of which this whole transposon can jump out from its original location and get itself integrated into a new location in the chromosome. When we come to non-composite transposons, you look here, see there is no insertional sequences present bordering the ends of DNA. Since these non-composite transposons do not have IS elements at the two boundaries, so they need to have their own transposase gene. So as you can see, they have their own transposase gene. They have this resolvase enzyme. This is a replicative transposition. So it is keeping a copy of itself and then moving out into a new area in the chromosome. So it has another enzyme, the resolvase. There's a third enzyme present. It's not associated with transposition phenomena, but beta-lactamase. And you can see on the two boundaries, there are about 38 base pair inverted repeats. So there is this left inverted repeat and there is this right inverted repeat. So composite so when the concept of differentiating between composite and non-composite transposons arise, obviously keep it in mind. You explain it with an example. So the example of a composite transposon is TN10, and the example of a non-composite transposon is TN3. I'll show you some more example of composite transposons. See, this is a non-composite one, the one without the insertional sequences at the boundaries. So the next pictures will show you some of the composite transposons. So you can see there are three more composite transposons shown here. We have this TN9, then TN5. TN9 again consists of two IS elements. TN5 also consists of 2 IS elements. TN10 also consists of 2 IS elements. So as you can see that these are composite transposons which have IS elements on the boundaries. To repeat, non-composite transposons are not do not have IS elements on their boundaries, but they have their own transposes gene which facilitates their transposition. Now let us have another concept or another perspective of transposition. Now, what is the new perspective of transposition? The new perspective of transposition is that autonomous and non-autonomous elements. The name itself suggests autonomous means self. So these DNA transposons have their own transposase gene. Since they have their own transposase gene, they can promote their own transposition. So that is autonomous. But non-autonomous means these are also DNA mobile elements, but they do not have the transposase gene and they have internal inverted repeats present on either ends. Somehow they do not have this transposase gene. So by now you have understood that if there is no transposase gene, even if these mobile elements are bounded by IR elements on the both sides, they will not be able to jump. So they are freezed in the genome. They have the capacity to jump only if somebody provides them the transposase enzyme. So when I'll be explaining you the Barbara-McClintock experiment of ACDS system, you'll understand this phenomena once more. But for now, keep it in mind that DNA transposons who have their own transposase enzyme can very well jump in and out. DNA transposable elements without their own transposase gene cannot jump. But if a neighboring transposon provides a transposase enzyme, then these DNA elements will be able to jump. So, as I said, that cell carries the autonomous transposon. Often cells carries having this transposase enzyme. So, if in presence of this transposase enzyme, the non-autonomous element will be able to transposase. So it needs for the non-autonomous elements, a helper transposon is required for it to jump. So with this now, we move on to, as I said you, that I'll be explaining the mechanism of transposition. I said we are going to do the cut and paste and the replicative transposition by animation. So I'll be moving on to the animation of the one provided with the molecular biology of the gene book. So we'll move on to now animation part. So now, as I said, we'll move on to this animation part and we will try to understand the two basic mechanism of transposition that we have discussed so far, the cut and paste transposition and the replicative transposition. So let's start. So the objective of this animation is to teach you to understand the cut and paste transposition and replicative transposition. This is the original DNA from which this, as you can see, the green portion is a transposon. So for all the animations that will be following, the green portion is the transposition, the gray color DNA is the donor DNA, the blue color DNA is the target DNA where this transposon will integrate itself. Now, as I had said, the recombinants responsible for transposition are usually called transposase. Now, transposons carry a gene encoding their own transposase. So if they're autonomous, they have their own transposase gene. Often, transposons are also seen to carry a few additional genes. For example, many bacterial transposons might carry antibiotic resistance gene. So when we are talking about a scenario like this, keep it in mind that this is an autonomous transposon. That is, it has its own transposase gene, which will give the enzyme transposase for it to move. And as I had told you, you can read it, the sequences, the very end of the transposon DNAs are arranged as inverted repeats. These inverted repeats range from about 25 to few hundred base pairs and carry the recognition sequences for transposase binding. So if I read the sequence 5 prime, TAA, TGC, GTC, again, the other strand 5 prime, TAA, TG, CG, TC. So these are inverted repeats. One of the basic criteria of all mobile DNA elements that can jump is that they are bounded by these inverted repeats. When they insert themselves into the target site, the direct repeats are generated. So this is a simple cut and paste transposition. As you can see, it is excised from its original location in the genome, followed by integration into the target site. Now, as you can see, this is a non-replicative mechanism because when it is cutting itself out from the original site and entering into the target site, it is not keeping any copy of itself in the original position. Now, what are the steps of cut and paste transposition? The first step is that the transposase enzyme or the subunits bind to the terminal inverted repeats. To recapitulate, all mobile DNA elements have terminal inverted repeats at their borders. So these two enzymes coming out from the transposase subunits, which are produced from the transposase gene of this DNA, mobile DNA element, is able to bind to the two ends of the inverted repeats. Next, you see, once the transposase subunit recognize these sequences, they bring the two ends together to generate a stable protein DNA complex known as transpososome or synaptic complex. This complex functions to ensure that DNA cleavage, DNA has to be cut, and joining reactions occur simultaneously on the both ends of DNA transposon. So you cut the DNA, you just cannot allow it to loosely roam about. So there's this cutting and joining reaction taking place simultaneously on both ends. And it also protects the ends of the DNA from cellular enzymes during transposition. So the first step was binding of transposase subunit. The second step was transposon formation. Final step is the excision of the transposon. So as you can see, what happened? The transposon is excised from its initial location in the genome. And to do this, each of the transposase unit, subunit, nicks one DNA strand. The 3' end is nicked. This is the green color part is the transposon. So the 3' end of each of the DNA of the transposable element is nicked, such as the transposon sequence terminates with free 3' hydroxyl groups at each end. So excision of the transposon, one strand of the DNA has been nicked. Now what happens? As you can see, to finish this excision reaction, the other DNA strand at each end of the transposon must also be cleaved. So different enzymes use different mechanisms to cleave this second DNA strand. For some transposons, an enzyme other than transposase directly cleaves this strand. So the other enzyme, another enzyme cleaves these strands. So both the strands have been cleaved and the mobile element is held in this position by the transposase subunits. In some cases, the transposase catalyze attack of the uncut end of the DNA strands by the 3' hydroxyl group directly opposite to them. Anyway, in whichever way the cut is done, you see that the two ends join. This two ends join to form the DNA hairpin. Once these two ends are joined but to form the DNA hairpin, the hairpin are subsequently hydrolyzed by the transposes. Some use different complex mechanisms to remove this hairpin, because since this is a 3' end and this is the 5' end, automatically there is a tendency of the 3', 5' to bond, and hence those hairpins form. So you need to remove these hairpins because this is now supposed to be integrated into a target site. Now DNA is transferred. So this is the target site, and you can see the 3' OH is attacking the target DNA. This reaction is called DNA strand transfer. As I said, the site of attacks on the two target strands are usually separated by 2 to 9 nucleotides. A staggered cut is made. And to emphasize on the fact that the distance, the sites of attack of the two target strands, the distance of the attack is usually fixed for each type of transposon. For one category of transposon, the target site is fixed. So once it cuts the DNA at the target site, you saw that the two DNA opens up. So it basically a staggered cut has been generated. Next, the gap in the target DNA resulting from the staggered cut on the two strand is then filled up by cellular DNA repair proteins. So it's being filled up. And these filled up areas are basically direct repeats. So as you can see, initially this was the green portion was the transposable genetic element. The light green was the central DNA part. The dark green were the borders, which are the inverted repeats. When it enters a target site and makes a staggered cut, and when this staggered cuts a field, you saw that in the target DNA, the direct repeats were generated. So these are known as target site replication. So this is basically in a stepway manner how a cut and paste transposition is occurring. To repeat, first the binding of the transpose is subunit to the terminal inverted repeats. Then we saw the transposon formation, excision of the transposon, DNA strand transfer, and then gap repair. So these are the 5 basic steps in which the cut and paste transposition occurs. Then you will be looking into the replicative transposition. The replicative transposition is a bit critical as compared to cut and paste transposition. Now, the beautiful thing about this animation of Watson's molecular biology of gene is when they will be showing you the replicative transposition mechanism, they will be showing it to you in a comparative manner. So there'll be a split window. On the lower half, you'll be shown the cut and paste transposition. On the upper half, you'll be shown the replicative transpositions to have a comparative analysis of how the thing goes. But this is just to tell one more thing, that the original site where the transposon was present in case of cut and paste transposition, since double-stranded breaks were initiated at the initial site of the transposition, so they must be repaired. And these breaks can be repaired by the homologous recombination or joined directly by the non-homologous end joining pathway. Now, as I said, to come into the replicative transposition general overview. So this is a split window, as you can see, the upper one showing you the replicative transposition and the lower one cut and paste transposition. They will go over it one by one, but keep it in mind before seeing this animation, the gray strands, once more I'm repeating this, otherwise we'll get confused. The gray strands are those DNA which has the original DNA transposon. So that is the donor DNA. The gray strands are the donor DNA. In both cases, the gray strands are the donor DNA and the blue strands are the target DNA. So in case of cut and paste, you saw that it was a simple cut and integration into the donor DNA. Here we are seeing some different but a critical phenomenon. Keep it in mind, the gray strands are donor DNA, the blue strands are target DNA. Keep this in mind. And another thing to be kept in mind, in cut and paste, no original copy of the transposon left in the donor DNA. In the replicative one, we are now having, so the number of copies of transposon has increased. This is important. Here, we started with one transposon, we ended with one transposon. The place changed. In case of replicative, we started with one transposon and we ended with two transposons now. So automatically, as you can understand, in replicative transposition, the number of copies of transposon is increasing. So it's a very similar manner. Initially, you can see the transposon subunit binds to the initial terminal inverted repeats, transposon formation. This is similar. Then we'll see the 3' hydroxyl nick at one end. So cleavage of the 3' OH generated in both cases. The difference here would be, if you remember, both in case of cut and paste, you can see both ends have been nicked off. But in case of replicative, it's only the 3' OH end that has been cleaved off. The other end is remaining attached to the other donor DNA strand. So this is one first difference that you should note. Here the transposon is separated from both the DNA strands. Here you can see there is only this 3' OH nick. The other DNA strand is not excised from its initial location. Now comes the concept of DNA transfer. So once you saw, what did we see? We saw that the 5' end of the transposon sequence remained joined to the initial flanking sequences. So these 5' ends remain joined. So when it is the 3' OH is targeting the phosphodiester bond of the target DNA, here what we see, one of the DNA strands of the target DNA gets attached to the 3' end and the other DNA strand gets attached to the other 3' end. In case of cut and paste, this criticality doesn't arise because the transposable element has totally excised itself out from the original donor site. I'm again repeating this particular step and see how this cutting is being done. So that's the target site. So the replicative transposition intermediate therefore consists of a doubly branched DNA molecule. You see? So we have got something like this. Now, this is important. What we see is something of a replication fork at the two ends. So automatically the donor and the target strands separate out. In between the gaps are filled. So this red color portions are the portions that has been newly synthesized based on the sequences present on the ssDNA. In case of the cut and paste transposition, these are direct repeats and this results in target site duplication. In case of the replicative one, you can see that there were two replication forks. The picture showed the presence of two replication fork on two ends, and those two ends behave as a replication fork, and taking the two single-stranded DNA as template, the remaining of the DNA is completed. The gap is filled. Once more, we go over this. So here the 3' ends of the knit target DNA, in case of replicative one I'm talking, serves as the DNA primers. And filling of the gap occurs, the one we saw just now. Now, in case of replicative transposition often rearranges the flanking chromosomal sequences. For some transposon, site-specific recombination occurs between the two transposons, resolving these rearrangements. So let's go over this once more. You see, in this position, this is the donor DNA, target DNA, donor DNA, target DNA. So they need to move back to their original position. So site-specific recombination is occurring. Copy of the transposon is present now in the donor DNA with a fresh copy of the transposon present in the target DNA. So with this, we come to the end of this animation, and also to the end of our class for today. So we'll stop our class today here.